So for the next 45 minutes or an hour or so, we're going to have a bit of a panel discussion here about cancer vaccines. And I'm Darrell Irvin. I'm one of the associate directors of the Koch Institute. It's my honor to get to moderate this discussion. I'll introduce our panelists first. We have Kyle Holen, who trained as a medical oncologist at Columbia University and Memorial Sloan Kettering Cancer Center, and began his career treating patients with GI tumors at the University of Wisconsin. He led early phase trials and novel therapies for GI and neuroendocrine tumors. He left academia to join AbbVie in 2009, and now he's currently head of development at Moderna, responsible for clinical research programs in oncology, rare diseases, cardiovascular, pulmonary, and autoimmune disease. Welcome. Bob Langer is one of 12 MIT Institute professors. He's a member of the National Academies of Medicine, Engineering, and Sciences and the National Academy of Inventors. He's led groundbreaking work in biomaterials and drug delivery over many years. He started many companies, including, for today's conversation, he's a co-founder at Moderna. And he's done a lot of research into technologies like lipid nanoparticles that are now important components for mRNA vaccines that are an exciting modality for infectious disease and cancer. And lastly, we have Kathy Wu, who's a professor of medicine and chief of the Division of Stem Cell Transplantation and Cellular Therapies at the Dana-Farber Cancer Institute. At DFCI, she oversees research and clinical activities focusing on dissecting the basis of effective anti-tumor immunity. And importantly, she's pioneered clinical trials of neoantigen targeting cancer vaccines, trying to move this modality forward for patients. And she's a member of the National Academy of Medicine and the Association of American Physicians. So can we have a welcoming round of applause for our panelists? I thought I'd start the conversation by setting some very recent context, especially for the trainees in the audience that may not be completely looped in on what's happening. It's actually a really exciting time for cancer vaccines, specifically as an immunotherapy in cancer. There have been several trials that have recently read out very encouraging data. One of these was a trial run by BioNTech in partnership with Genentech in pancreatic cancer patients, where they sequenced patient tumors and prepared mRNA vaccines, patient-specific neoantigen targeting vaccines that were administered post-surgical resection in those patients alongside checkpoint blockade and chemotherapy. And that trial is 18 months of follow-up. The patients who responded to the vaccine have essentially zero recurrence of the tumors so far, whereas non-responders are recurring as expected in that lethal disease. So that's exciting. That work was published in Nature just in the last few months. Pete DeMuth's going to talk about some exciting, from another small trial in mostly pancreatic cancer patients, a KRAS targeting vaccine. He'll talk about that this afternoon. And then, Kyle, I was hoping maybe you could comment a bit on the exciting data coming out of Moderna's trial in partnership with Merck.
Happy to do so. Would you like me to talk about it now?
Yeah, just briefly to sort of set the stage here, I think.
Sure. So mRNA 4157 is our individualized neoantigen therapy, which is similar in the same process as the BioNTech vaccine, where we sequence the tumor, identify the neoantigens, and then create the mRNA that's individualized to that patient. And we just released data both at AACR and ASCO showing 44% decrease in the risk of recurrence in high-risk adjuvant melanoma patients, as well as a 65% decrease in the distant metastasis-free recurrence rate for that patient population. So we're very excited about those results and can't wait to see whether or not it has similar effects in other tumor types.
Yeah. So a lot happening very recently in the clinic here. But now I want to back up because there's a prominent editorial almost exactly 20 years ago written by Steve Rosenberg and Nick Restifo, who are leading cancer vaccine trials and other immunotherapies at the National Cancer Institute. And they compiled in 2004 the collective data from a whole variety of cancer vaccines they had tested in patients, in about 400 patients, and they found they had a 2.6% objective response rate. And so they had written this editorial to say, we need to do something different. And I wanted to ask you, Kathy, what do you see as the major steps that have happened from that time to now, where we're seeing these exciting things happen?
So I read that article, and it was a major bummer. So I think I was kind of towards the end of my postdoc and trying to forge a career in this area, and to have luminaries in the field kind of say, it really isn't working. It's just kind of like, okay, what are we doing here? But I think if you look back to that experience, and you go back, and this was published, I believe, back in 2004, and you consider what was going on in the field at that time, there are many reasons why you could think that the response rates were pretty dismal. One is just the maturity of where the reagents were. Another aspect is that a lot of those trials were targeting single antigens, and we know so much through all the TCGA efforts and all the collective genomic characterizations that we've done of cancers over the past decade. We know that cancer heterogeneity is alive and well. So if you're just going to go after a single antigen, then you're destined for failure. So I think that has to be said. And then I think, but if you actually look a little bit more deeply into that experience, there were some hints and murmurs of success. And I am a glass half full type of person, so let's just say that. But the more complex formulations seem to have some sort of activity. And by complex formulations, I would say the whole tumor cell vaccines, in particular, seem to have a little bit of activity that almost begged wanting to kind of optimize and hone into that aspect a little bit more. And I think this kind of leads the conversation to where we are now, where we're trying to think about multi-pronged targeting of antigen. In terms of what has happened over those 20 years, I think first, I would say a number of very important technological things. I think the concepts have always been in our head, but how do we actually get there? Next generation sequencing, for sure. That I think that has made tumor neoantigens a reality, because it's provided a tractable, feasible, cost acceptable approach to scan an individual tumor exome or genome and actually pick out the candidates. I think the other aspect that certainly by the time my group entered the field, I was gratified to see that our HLA binding predictive algorithms had moved beyond HLA-A2. So certainly when I was a postdoc, we were all obsessed about HLA-A2. And by the time next generation sequencing was coming along, there had been already a number of competitions between different tools and algorithms, so that there was a winner, that these neural network-based approaches seemed to be providing enough specificity and sensitivity to get us somewhere, and that there had been enough mining of the data so that the models could be applied across different HLA alleles. So I think those are two aspects. And then we heard from Federico Sallusto this morning that we've come a long way in terms of trying to think about T cells and their specificities, and we just have so many great single cell tools right now that allow us to now really dive into the responses and try to identify individual T cells and their specificity. So it's a progression.
But so many things in the immunology. And then also, I think, on the technology front, much better tools to vaccinate. And it seems like mRNA is really a key, at least one key way to make personalized vaccines possible. Kyle or Bob, if you wanted to comment about, how RNA makes it possible to make a patient-specific vaccine.
Oh, sure. It is challenging. It's not for the faint of heart to create an individualized therapy. But it comes with, I think, a wealth of opportunity. And what I'm particularly excited about with the RNA platform is it is a learning product, which I don't know if we've ever experienced in pharma before, where you can train your algorithm that's selecting your neoantigens and learn over time and have new algorithms 2.0, 3.0, 4.0 that can just improve our selection process for neoantigens. And that's super exciting for me to know that this isn't just a product that is going to be the same over time. It's a product that can continue to understand where it's making a clinical impact and take that information, feed it back into the algorithm, and just get better over time. Now that creates a little bit of a regulatory quagmire, and so we haven't figured that one out yet. In fact, even our manufacturing process has been a novel concept for our regulatory colleagues around the world, because the algorithm is part of our manufacturing process. And so it's something that has to be viewed along with our typical release assays and our all our manufacturing processes. So it's a challenge, technical challenge for sure, but one that comes with a wealth of opportunities.
And the other element is the need to manufacture this very quickly. And I think BioNTech reported 9 weeks to make their vaccines. This is something that wouldn't be possible with a recombinant protein-based vaccine. What do you see as the future of this? How quickly do you think we can make these vaccines?
Yeah, we're at about 6 weeks now. We're trying to make that even faster. But there are limitations that it's gonna be hard to get around. One of those is just shipping material back and forth. Now Amazon has gotten pretty good at that, so maybe we can learn a few tricks from overnight shipping from them. But even just getting specimens out of pathology labs is a bit challenging. So there are logistical challenges, and there are just technical challenges around the length of time it takes to create our plasmid and the length of time it takes to make sure we have the right release assays in place. So there's things that I think will be limiting, where we won't be able to do a one-day turnaround ever. But we're doing our best to try and shorten that timeline as much as possible. And I think in some settings, it's not that important to shorten the timeline. So as an example, in the adjuvant setting where we are working right now, there's a recovery period after surgery where people really can't receive treatment until they recover from their surgery. That's a perfect opportunity for us to go to work and create their 4157 for them, so that we have it ready at the time that they're recovered from surgery. So it really depends on the setting, and the timelines I think will get better, but there will be a limitation in how much better that can get.
I think that, and the other sort of technological important advance that didn't happen overnight was the development of these delivery materials. And Bob, I was hoping you could comment a bit on the progression over the past 20 years that have gotten us to these multiple approved mRNA vaccines and new applications every day.
Sure. Well, this really goes back many, many years, and many groups contributing. I mean, so we started trying in our own lab back in 1976, well, 1974, but published in Nature in 1976, using materials like polymers and other things to see if we could encapsulate and deliver large molecules. you know, people didn't think that was possible, and actually even after we did it, I think my first 9 grants were rejected because of that from the government, NIH. But then, you know, a few years later, there were some other papers that would lead to where people not only, so we encapsulated nucleic acids and proteins and so forth, then and delivered them. Then there were, but even in spite of those obstacles, then within a couple years, several other groups had done this in what are called liposomes, which is a precursor to lipid nanoparticles, I suppose. And they actually formed a company called the Liposome Company. I mean, not to do that, but to deliver small molecules. Just to take a step back, you know, if you look at what's a lipid nanoparticle today, there's usually 4 components: cholesterol, some type of structural lipid like a phospholipid. Both of those were in the liposomes. But the other 2 things that were important is adding some. So if you make these tiny particles, there's at least 2 issues. One is that they can aggregate, you know, because they're so tiny, so they can stick together. There's so much surface area. The other is that, you know, when you inject them, macrophages or other cells can eat them. So a way to prevent that, and that was done by Leaf Wang, Vladimir Torchlin, and also our research group, is to add what's called polyethylene glycol, which is actually in Miralax. If anybody needs to go to the bathroom, it's the same thing. But at any rate, I'm not necessarily recommending that. But at any rate, but so that, and there was a number of papers in the 80s and early 90s on, you know, like by those groups, including ours, to show that. Then another, I think, key aspect was Phil Feldner's work in 1987. He was at a company and showed, and so the problem was that, you know, to try to condense the nucleic acids and to make them small enough, and so he made what's called cationic lipids. That's how a lot of this started. So, but the problem with the cationic lipids is that they could cause complement activation, you know, they could cause toxicity, and they could cause, you know, sometimes having a hard time to get out of the endosome. So actually in 2000, Dan Pack and David Putnam, who were in our lab, Dan came from Francis Arnold's lab at Caltech, and Dave came to the lab. So they, using polymers, had this idea of what are called ionizable groups. And the idea of that is that if you're, that when you're at 1 pH, it's going to be neutral, so it won't cause these problems. But when it gets into the cell, you know, then it can become more cationic and disrupt the endosome. So that hopefully would get rid of the toxicity problem. And the next year, Peter Cullis at Vancouver showed that he also came up with these ionizable lipids. But one of the issues with the ionizable lipids is, you know, lots of them didn't work that well or were toxicity. So actually, Alnylam was started by Phil Sharp, who was at our institute, and others. We collaborated. Dan Anderson, who was then a postdoc in our lab and now also at the Koch Institute, came up with the idea of making these giant libraries, so you'd make thousands of lipids. And that would give you much more availability. And then there were also manufacturing challenges, some which Alnylam worked on, some which actually Rohit Karnik, and who's also a postdoc with us now, professor at MIT, developed, and other companies as well. And then you go to, you know, like 2010, and now you have a number of companies working on mRNA. Moderna had started, and BioNTech, and those companies, you know, you wouldn't necessarily use the same lipids or the same lipid nanoparticles. So again, so what happened is there was a company, Acuitas, and they had worked with Alnylam you know, early on, and they worked with BioNTech to come up with the right kind of lipids. Again, making large, not the libraries the size that Dan made, but a lot of the Moderna did something similar. It may depend on not only the size of the molecule, but also whether you're going to inject sub-Q or IM or IV in terms of what you're doing. So I think there were many, many different groups that made, you know, lots of contributions for many years, and now, and there's still long ways to go. I mean, I think that, you know, trying to make them more shelf stable, trying to target them to different cell types, I think there's a lot that will continue to go on.
Yeah, great. I suspect it's no accident that these recent trials are going to a specific setting of patients who've had surgical resection or in some type of sort of minimal residual disease state, and we're combining it with checkpoint blockade in some of these trials. What do you see as the importance of picking the right trial design, and where do you see us going next?
Yeah, no, thank you for that. I think that's a really important question. I think anytime you embark on a novel therapy, you want to make sure that you select your clinical setting very carefully, because you have to keep attuned to what is the question you're asking and whether you're actually going to get a informative result as a result of the trial. And so I think that I don't think it was by chance that the first personal neoantigen vaccine trials that came out between our group at Dana-Farber Broad, MGH, and then Uğur Şahin's group in Germany, I mean, these were both in the same clinical setting, which was adjuvant stage 3, stage 4 melanoma. So you want a disease that has a known high risk for relapse, and yet these are patients who, for a cancer patient, are sufficiently immunocompetent, so you think that you can mount an immune response. A vaccine requires something of the patient, which is the ability to mount an immune response. So we didn't want to design a lengthy, something that would take a long time to design only for the patient to progress or require chemotherapy or something else that would confound what was going on. And for this type of therapy, at least kind of for the first proof of concept, we wanted to have broad latitude in terms of the targets that we could select. And it was well known by then that melanoma had a high mutation burden. So that made it a suitable setting. I think going forward, I think we would all acknowledge that the immune system works better under conditions of minimal residual disease or absent disease. So I think vaccines are really great either in early disease or following surgical resection or following treatment that renders them, at least at that time, disease-free. So thinking about settings of minimal residual disease, I think some of the recent exciting trials that are coming out are doing exactly that. And then adding co-therapies such as immune checkpoint blockade that can actually take those soldiers that you've mobilized and actually getting them to full throttle function is also really attractive. So I think many of us can think of different cancer settings where there is a minimal residual disease state. I think contrary to kind of the traditional paradigm in novel drug development, we should not be going to kind of the worst, the most advanced disease, metastatic, where, I mean, that's not the right setting for this.
Yeah. And Kyle, of course, Moderna's pursuing many different therapeutic strategies. So where does vaccination fit in the overall strategy for oncology at Moderna?
Well, it's a big part of our strategy, given the exciting results that we've observed now for 4157. I completely agree with everything you said about the setting. Just want to add that one of the challenges with these end-stage refractory metastatic tumors is they can lose their ability to even present antigens with downregulation of HLA and other. So you just, I think there's more of a likelihood of immune evasion if you're going into really late stage settings, which is what we wanted to avoid. We also wanted to pick a tumor that had a history of responsiveness to immune manipulation, and clearly melanoma is high on the list. So those were all part of our selection, and will be part of our selection for our next stage studies. But beyond our vaccination approaches, We also have approaches where we are administering mRNA to secrete specific antibodies. So using hepatocytes to secrete bispecific antibodies and other antibodies that might help modulate the cancer cells. So there's a variety of different ways that you can use mRNA therapies outside of a vaccination type approach, and so we're pursuing all of them.
Do you think that that some success in the current settings and using these personalized vaccines may open up the possibility to do more in the vaccine space? Are there prospects for off-the-shelf vaccines in any setting, preventative vaccines, where Olefin's going to talk about preventative vaccines? Do those maybe more difficult trials in that scenario become more palatable if we start making headway in the established disease?
There's lots of parts to that question. So, I mean, I'll take a stab at it, and
Sure,
I mean,
please.
I think vaccines. I think we would all agree are an important adjunct to a lot of different cancer therapeutic modalities. So I think that it is a very good partner to other immunotherapies that have signal. I think that a lot of our efforts, and I think the efforts in the field, have been focused on therapeutic vaccines. So patients who already have cancer, how do you get rid of the cancer? And I think that vaccines by themselves, I think it is a big ask for the vaccines by themselves to do everything. I think some of the recent studies that we're hearing between Moderna and Merck, and also what the experience from Memorial Sloan Kettering, this is vaccines together with other modalities that have rendered the cancer cells more vulnerable to the vaccine. So I think as a single entity, I think it's hard to use unless you're really in a prevention sort of approach where the cancer has not started yet. That will require all what we talked about before, which is improvements in cost and scale to which vaccines can be generated, because you're going to have to be able to reach a large proportion of the population and rapidly. So this is like what we did with the COVID vaccine. So I think, and that opens the question as to what do you vaccinate against? So that's a whole nother big subject. And I think as we start to think about where we can really focus on cancer prevention, we have to ask ourselves, what are the targets that are reasonable that one would go after? Again, those kind of delivery and technology parts are really enmeshed in that whole question, because I think there's going to be safety together with cost and timing.
I'll just add, I mean, you said it well, there's not much to add other than I think if we're going to use an off-the-shelf approach, you better be pretty confident about the target that you're selecting and why you're selecting that target, and whether or not there are combination strategies that can also affect the target together, and the importance of that target as an immune driver, as a cancer driver, cannot be overstated. So I think, yes, we are pursuing off-the-shelf strategies. We are not pursuing them as monotherapy or necessarily as a direct effect on the cancer, but perhaps more of an effect on increasing an immune responsiveness to the cancer. And also in combination with other agents such as small molecules or biologics or cell therapies, where we know what the target is that these cell therapies are going after. Can we help the T cells attack the cancer by using a vaccination approach? So I think in that setting, an off-the-shelf vaccine is being used more as an adjuvant to a therapy that might be working well but not well enough.
I think we'd like to take some time to open it up to let the audience, if any of you have questions you'd like to ask the panel as a whole or any of our panelists, please come up to the microphones.
Thank you for your inspiring discussions. So can you comment on the perspective that we develop an ensemble of vaccines and RNA medicines and nanoparticle-based delivery systems targeting a community of cells, including B cell, NK cell, macrophage, besides tumor and T cells, in order to educate the tumors, not only eliminating the tumor cells?
Can you rephrase?
So I think if I got the question right, sorry, because the acoustics up here are really terrible, that you're asking what cells should we be targeting with these vaccines?
So we can target a community of cells,
A
all
community of
cells.
cells.
Yeah, what should we be targeting multiple cell types for vaccines?
At some level, I mean, I think this is achievable through different delivery approaches, and obviously, if you start to add different bells and whistles that can, as you said, bring a community together, I think a healthy immune response does require multiple different arms of immunity, and at least in our group, that was the rationale behind going after synthetic long peptides, because at that time there was a lot of data to help support the notion of bringing multiple different T cell subtypes together. I think with mRNA there's a great deal of flexibility in terms of how it can be constructed so that you can attract many relevant cell types to the site of vaccine. So I think you bring up the important concept that robust responses do require multiple heterogeneous cell types. And I think the second concept is now how can we actually actuate that in the patients that we treat.
Both from an mRNA perspective and the delivery mechanism, the LNP,
Yeah.
both important in making sure we target the different cell types. So yes, I think that's
And
spot on.
Bob, you've done many proof of concept studies suggesting that it's not beyond the realm of possibility that we have LNPs or other materials that can get these nucleic acids to specific cell types in the body.
I'd say that that's right. It's not. It may depend which specific cell types, but that's like an ongoing challenge. But to some, we've certainly been able to do that. And I think over time, there's going to be again more and more success in that area.
The answer is yes.
So I want to just comment. I was coming to this meeting today hoping that I would hear about a universal antigen in a lung cancer of a certain type in a certain state of development. And that hasn't emerged. In fact, well, I hope you tell me.
Yeah, it's
But
coming
then-
in the afternoon, Phil.
Come the afternoon. We'll wait till the afternoon, but I can ask a question now. The creation of a personal antigen in this context is treating a patient of one, because you sequence a patient ideally and you create a vaccine and you're treating a patient of one, because the next patient may call for a different vaccine. Patient one raises many challenges. The classical challenge is how do you prove toxicity? How do you, if I take an RNA vaccine or an RNA to patients, I test it in mouse, in primates, other animals. I go through a toxicity test, and then I go to an efficacy test, and a general utility test. Patient one doesn't do that. So it's almost an FDA issue about a patient one. A universal antigen you can test in a variety of different ways. So if it's a patient of one and you're using a complicated algorithm, does the algorithm need to be disclosed as part of the therapy for a patient of one immunization?
Well, I guess I can share with you a little bit about the algorithm. So we haven't disclosed the algorithm to each patient, if that's what you were asking, but we have disclosed this to regulators so that they know how these antigens are selected. As I mentioned, it's part of our manufacturing process, and that is something that regulators care deeply about to make sure that we have consistency and quality measures in place for our product. So yes, that algorithm is something that regulators review as part of the process, the review process. But you're right, there are challenges for sure. We have 34 different neoantigens that are unique for every patient, and so far we haven't seen anything that suggests that any of those neoantigens create a safety issue that's outsized from the next dose that we administer. But I will admit that we have 200 plus patients that have been treated so far, so we don't have a wealth of information. But very good points.
Maybe I'll just add a few comments, because I think, and I'm kind of locking eyes right now with Ed Fritsch, who's in the audience, because we were on the team that kind of went down to the FDA back when we first opened our first proof of concept clinical trial. And I think that we found the FDA's very collaborative, very receptive. What we presented was a weight of evidence that supported the idea that these were truly tumor-specific targets. Their primary concern is safety. There had already been work, of course, we were presenting for long peptides, and there was a weight of prior studies in humans that demonstrated the safety of pooling together even up to 15 peptides without adverse events. So that was already there. It didn't really make sense to go to preclinical models because by virtue of a personalized product, it didn't make any sense. And they did, in our regulatory filing, we did state our algorithm. They want to know that it's a rigorous process, that there are processes to ensure that there's reproducibility in terms of how you do your sequencing and the approach that you're taking to analyze the data.
Cool. So a previous question was about targeting specific cell types, but I'm wondering if we made any progress thinking about targeting specific tissues or lymph nodes. And I mean, I think a lot of these trials have been in melanoma, but if we move to pancreatic cancer or colon cancer, where are we even injecting? What additional, I guess, molecules can we add to the vaccine to imprint the T cells to home to specific organs? I mean, could we be diverting a productive T cell response by priming this massive response centrally, but not in the organ? And also related, I guess, to the strength of priming a T cell response, are you thinking about ways to modulate kind of the sweet spot, right, where you're going to be driving not this short-lived effector response that's going to burn out, but something that's kind of in between and it will have a large memory progenitor pool. Are we there yet, or is this kind of far in the future?
These are the questions we ask ourselves all the time. And I think there's so many aspects to vaccinology. I think hopefully this is comforting, but I think one aspect is that we hoped by kind of locking down on a good antigen, such as neoantigens, that would at least tack down one aspect of the question, and then you could start to iterate around the other age-old questions of vaccinology. So we do ask those questions. I think that, we heard from Tyler Jacks earlier with preclinical models. I do think that that is a wonderful approach to iterate quickly because we just can't do it fast enough in our human patients, short of doing small directed trials that are just focused on asking that particular question with not many patients but informative patients. Then it becomes a matter of funding and how do you get that set up. But I think some iteration between preclinical models and patient data hopefully will help us with those type of questions.
And I'd add here that this is also an area where there's still a lot of exciting, important work happening in basic cancer immunology about the status of T cells that respond to checkpoint blockade, that mediate cancer regression. Tyler mentioned these stem-like T cell populations that we know nothing about how to control and induce those by vaccination that might be key to making the next step forward. So.
You could argue whether or not an IM injection is adequately getting to the antigen-presenting cells, or whether maybe an IV injection might be better in getting to the spleen, and that maybe there's a higher population of antigen-presenting cells. So I think those are some questions I think are yet to be answered. In terms of targeting specific tumors in different organs, I think where that maybe is the most challenging is when you're thinking about GBM and brain tumors, because there you would hope that T cells could get to most of the organs inside your body, but there is the blood-brain barrier where I'm not as certain that we'd get the same robust T cell populations into the brain in order to have an effect on brain tumors. So that is yet to be seen. And so that's where I think I have a little bit of a concern around whether or not we can get adequate T cell populations to be effective in a target organ. But outside of the brain and maybe the testes, I guess, I think we should have, as long as we have a robust T cell response, we should be fairly confident that it's going to get to the target organ.
Cool, thank you.
Great discussion, thank you. So what about cancer vaccines, like efficiency for necrotic cores and solid tumors was my question.
In what, sorry?
Necrotic cores and solid tumors.
Yeah, I think it's another reason why we've been hesitant to jump into the metastatic setting with large tumors where you have inconsistent perfusion across the tumor. Certainly you don't have any blood perfusion in the necrotic centers. So I would like to explore that for sure at some point and see whether or not we'll have activity there, but it's something that we'll need to tread lightly. Let's see if we have proof of concept in the adjuvant setting first before we jump into these bulky tumors with necrotic centers, because I think it will be a challenge for sure. Any thoughts on your side?
No, I was just actually I was just reflecting from the questions and thinking about one of your early questions, what has happened in the last 20 years, and we mentioned devices, formulation, sequencing, algorithms, but I also want to emphasize that we're in this incredible time of having the tools to actually dissect immunology in patients. And I think that's actually what makes it so rewarding to work in these different patient settings and try to get at the questions that one of the earlier participants asked, because we can directly go to patient specimens and actually start to pull out all these different pearls and I think that's a very new thing over the past decade.
Thank you.
Hi, thank you for the stimulating insights and the future prospects in the cancer vaccines. I'm interested to know more broadly what would be could Bob or anybody in the panel could comment, what would be the future in use of biomaterials and immune system moving forward, and how can you pick and choose the questions to address not the biomaterials and the immune system in itself, and also the second part of the question is if you could give an advice for young researchers like me looking into immune system and biomaterials, how would you bring different technologies? For instance, recently I learned about a review on vaccine storage, how it is important to maintain the cold chain supply, so that you can efficiently distribute the vaccines without compromising their efficacy, especially in low and mid-income countries. Could you comment on this? Thanks.
Well, there are a lot of questions in there. I'm not sure I can answer them all. Maybe I'll just make a couple points. So I think the lipid nanoparticles are I think there's a lot of improvements. I mentioned some of them that could be made before, and there's also other materials. I mean, like I mentioned, like we and Dan Anderson and others, and Paula Hammond. People are also doing a lot with polymers as another biomaterial. And I think there's, again, depending on what one wants to do, people have also looked, for example, at different structures, like not just nanoparticles, but micellar systems. There's several groups in Japan that have done a lot of interesting work on that. And then again, to the point that was made earlier, you know, there's a desire, I think, if we could get design materials that could, say, cross the blood-brain barrier and maybe other barriers too, better. You know, I'm trying to think of all of the other questions, but I think you also, you said, what advice? I mean, I just think, you know, I think it's a good thing to try to answer really broad questions, you know, that can have a big impact. I think that, you know, you heard from people all day on doing that, and you'll hear more in the afternoon. But I think to try to come up with both a better understanding of the immunology, better algorithms, I'm sure will make contributions, and as you pointed out, you know, improvements in materials. But I mean, just understanding things better and making things better and faster. On the cold chain issue, I think that also gets into biochemical issues. There's understanding the fundamental mechanisms of, I think there's a couple ways you can go about it. You know, understanding the fundamental mechanisms of inactivation and adding, from understanding that, maybe doing the right chemical modifications or adding the right excipients. Some groups have tried to put the vaccines actually in a glassy state. So a lot of times what causes these problems are moisture. So if you could prevent moisture from getting in or make it very tough, that's another kind of strategy. So there's a lot of different things that can be done.
Earlier the panel was discussing a little bit about patient selection. I think the ideas of going prior to HLA loss and maybe looking in the MRD space. And I wanted to ask the panelists to just comment. I think it was so intriguing to me in the melanoma data with the checkpoint and the mRNA vaccine that when MRD was looked at through ctDNA, there was actually benefit in both the patients that had MRD as well as those that didn't. And I guess I naively might have assumed that maybe all the benefit would have only gone to those with the ctDNA positive MRD state. So just thought it would be interesting to hear the panel's thoughts about patient selection and the use of MRD assays like that. Maybe we don't have good enough assays yet, or how do people think about it?
Yeah, you know our data well, apparently. Yeah, we were really excited to see and surprised somewhat to see that 4157 showed benefit in both the ctDNA negative and ctDNA positive patients. Particularly because the ctDNA positive patients, I don't know if you happen to see that recurrence curve that we showed at ASCO, but they recurred very, very quickly. So quickly that if I had seen that curve before we ran the trial, I would have excluded them from the study because I wouldn't think that we'd have enough time to get the vaccine on board and have an effect before they had their recurrence. Now the numbers in that population are quite small, so I think we have to look at that data with suspect for sure. But there was a significant improvement with the addition of 4157. So that, in my opinion, starts getting me wondering whether or not avoiding the metastatic patients is the right call, because essentially the ctDNA positive patients are metastatic. They just maybe aren't bulky enough for you to pick up on any type of imaging studies. So we should probably start exploring what type of metastatic settings we'd like to do as a proof of concept, just to make sure that that's not something that a vaccine approach would make sense. But for now, I think the ctDNA is really important prognostically. And so making sure that we have the right balance of patients in our studies who are positive and negative is going to be critically important. But whether or not we use it as a selection tool is a different story for sure. Any thoughts from the rest of the panel?
No, I agree.
Yeah, I think you just answered partially my question about ctDNA. And since mRNA is such a new modality, how we compare it with our previous modality that we put into melanoma patients, such as checkpoint inhibitors? Any biomarkers that we're looking uniquely with this type of modality? And as well, are we seeing a T cell expansion, T cell repertoire change in the patient's PBMCs as well as in the tumor biopsies? I know we're still trying to mature the data, but I don't know if you guys have any comments on that.
Well, I can share with you that we are actively exploring this, and it's very early days, but we're looking at a whole variety of different signatures, perhaps, that could help us predict which patients might benefit the most from mRNA, and I think the jury is still out. I don't, unfortunately, have any data to share with you right now. We're still doing active work in this setting. I believe we will probably be able to find a signature that can predict the patients that are more likely to respond. And that, by the way, hopefully will help us understand how to improve our algorithm so that maybe then the signature will change over time and be a different type of signature that we can then use again to improve our algorithm. So all this work is critically important, not just to make sure we have the right patients selected, but to make sure that we improve our algorithm over time and help a broader population of patients.
Great.
So I'd like to expand a little bit on Kathy's comment that we're learning so much in these trials about how the human immune system interacts with the tumor. I've been a card-carrying cancer immunologist for over 40 years, and we have been accused many times that we do work in cancer that basically cancer has done more for immunology than immunology has ever done for cancer. And I find us in that place right now. And there's nothing wrong with learning new things. And we are learning a lot of new things. And I think investigator-initiated trials in academia are, absolutely a good place to do that. But we have colleagues from industry who are trying to make a product. And this is where I get stumped. For example, the latest trial was published 16 patients, or started with 30, ended up at 16, or whatever the numbers were. Every major medical center in this country sees hundreds of cancer patients a year of every type, right? Will they all send you tumors to, create personalized vaccines, or who does it? Do we just, if we are going in that direction as a product rather than as acquisition of knowledge, do we not tremendously expand unequal delivery of care? I am personally from Southeast Serbia, where lots of people have cancer. They haven't yet even gotten the checkpoint inhibitor to arrive there. Are they going to send you the tumors? So how do you actually reconcile, that direction of personalized medicine, personalized vaccines, unique antigens. And by the way, just a plea to all the scientists, everything that the immune system sees for the first time is a neoantigen. What we're talking about here is mutated neoantigens, and it would be very good to be precise because there are shared neoantigens as well. So I just, I get very worried that we are going in the wrong direction and diluting our efforts that could go in a different direction that would treat more people more equally, provide more relief.
It's an excellent comment, and look, access to care is a horrible problem. You don't have to look beyond the city of Boston to recognize that there are people that just don't have the right access to care. I don't know how to solve that problem. I wish I did. All I can do is work with my scientists to figure out what we can do to solve the cancer problem. And it may mean that we have a drug that at first is expensive, but I hope over time we can get the drug to costs that are more reasonable, I guess. But I'm not in the commercial organization, so you shouldn't probably be asking me these questions. But I do have hopes that we can learn from these studies and figure out a solution to make sure that everybody has access to a treatment that will help them.
I think science is incremental, and I think there's different chapters to what we're doing, and I think that you know, the neoantigens and the sequencing and the algorithms.
Mutated neoantigens.
The mutated neoantigens, correct. I mean, you know, we've gotten to this point where seeing through a vision of whether or not they've been even targetable. I mean, Bob talked about his NIH grants. I think the first grant that I wrote on neoantigens, one of the comments is, what is the point of this? This will never go to clinical trial, which of course made me go, just watch me. So, but you know, I think we are in a moment of discovery. I think what's so exciting about this year is that we're seeing coming to fruition large swaths of patients who are getting treated and being able to, the kind of questions that was asked about biomarkers, we couldn't have done that in our academic trials. The numbers are just too small. But I think with these larger industry trials, we can start to ask those questions. I'm pretty confident that we'll get to the next chapter, that we're going to be able to pay it forward with the knowledge that we're learning now, keeping in mind all those questions that you're asking so that we can actually get it out there, because vaccines are very important.
I guess I'm asking, what is the next chapter?
We have to figure it out together.
I don't think anyone knows.
Answer.
No, but it's true,
I agree
right?
with you. I
It's
agree
true.
with you.
I mean,
Nobody knows.
I think we gotta
We're
feel
learning.
through.
I mean,
Yeah,
that was the,
yeah.
Okay, great discussion. We're going to take two more quick questions and then we'll wrap it up. Go ahead.
Yeah, I just wanted to know if there were any comments on sort of the timing of the cancer vaccine when you're thinking about it's a combination therapy, right? You have your checkpoint blockade, sort of like a PD-1 that you're doing the clinical trials with. So where does the timing of the sort of cancer vaccine come into play? Because you're typically getting that sort of the checkpoint blockade every 2 weeks or something like that. So where are you then doing the adjuvant, right? So that's sort of my question.
I think it's a great question. Another question that we ask each other weekly in our kind of collaborative meetings together. I think there have been preclinical models that have suggested that timing of vaccine in relationship to checkpoint blockade does have an impact. At the Dana-Farber, we're currently conducting a trial to actually address that question, and we have different arms to look at the scheduling. I'm constantly reminded by my clinical colleagues that we have to keep a close ear to the ground to what is happening in the real world practice of our patients. And the reality is a lot of our patients are already on immune checkpoint blockade by the time they're coming for treatment. So you just have to realize that, and then whether or not preclinical models are a direct translation to the patient responses, it's similar but not always one-to-one. So
it's a great question, and we have no answers either. We just take a pragmatic approach, as you're doing, and understand, okay, where are checkpoints being used, how are they being used, what's the schedule, and where do we think 4157 might best fit in, using whatever preclinical data that we might have to help guide us, but it ends up ultimately being more of a pragmatic decision, unfortunately, as opposed to a scientific one. And I think we're going to have to design clinical experiments to figure that out. Thank you.
Awesome. Thank you all for being here today. It's been very educational. I guess the question I have is this, right? Recent data has shown non-traditional subset of T cells, like the gamma delta T cells, to be a good positive prognostic population in solid tumors. Like, what is kind of holding us back in terms of having more cancer vaccines to target these non-traditional subsets that are, you know, MHC independent, but also can be antigen specific as well?
Well, if you think we don't know a lot about normal effector T cells, ask us about gamma deltas. I think it's an exciting idea to think about broadening, you know, the arms of the immune system we're trying to leverage here. I don't know, but that's also still, I think, the cutting edge of where the science is going. I don't know, Kathy.
Look, it should be explored for sure. It's not something that we're actively pursuing right now, but I think it's something that is a novel concept, very interesting, and should be explored.
Thank you.
Thank you.
Okay, thanks everyone for a really stimulating discussion. Can we have thank our panelists one more time? And now we'll break for lunch. Enjoy your lunch, everyone.
Did a great job.